Hello viewers, in my previous lecture I had discussed sphingolipids with you. That is, I had introduced you to what is the basic structure or the backbone of sphingolipids, which is sphingosine. We have discussed sphingosine in detail, and we've also discussed ceramide, which is the basic unit of sphingolipids. I had also discussed with you that sphingolipids can be divided into two categories on the basis of the group R attached to ceramide. So, as I discussed with you in my previous lecture, the structure of ceramide, and you know that ceramide is sphingosine which is attached to a fatty acid at the second carbon, which you can see on your screens also. So now when we replace this H, I will refer to this H as R1. With the R1 group, we get different derivatives of ceramide. And as we've seen in our last lecture, that if this H group is a phosphoester group, then that is an example of the simplest phospholipid, that is sphingomyelin. Now, depending on this group R1, sphingolipids can be divided into two subcategories, that is sphingophospholipid and glycosphingolipids. And in sphingophospholipid, we've already discussed sphingomyelin in great detail. So when we talk of glycosphingolipids, so what is the difference between a glycosphingolipid and a sphingomyelin? You have to remember that glycosphingolipids do neither contain a phosphate group nor an amino alcohol. So what do they contain instead of a phosphate and an amino alcohol? They are attached to an oligosaccharide. And this oligosaccharide is directly attached to a ceramide by an O-glycosidic bond, which functions as a polar head. So when we compare the structures of sphingomyelin with sphingoglycolipid, what we find that in sphingosine as well as that in sphingomyelin as in sphingoglycolipids, the basic unit is the same, that is, it is the ceramide unit, right? This is the basic unit. But in sphingomyelin, we have the OH is attached to a phosphate group and a choline. So we have an amino alcohol here. Whereas when we will look into sphingoglycolipid, what we'll find is that this H here, or I'll rather say R1, H will be replaced by an R1 group, which will be a monosaccharide or a oligosaccharide. So you don't have to get confused that what is sphingomyelin, what is sphingoglycolipid. You have to just remember the basic structure of sphingosine, and then we will of course we will replace this H group with some other group like phosphorylcholine in sphingomyelin, and with a monosaccharide or disaccharide or oligosaccharides or some complex oligosaccharides, which will give us glycolipids. So if a difference is asked between a glycolipid and a sphingomyelin, what you have to remember is sphingomyelin is a phosphate lipid, whereas glycolipid has a carbohydrate head. Right. Now, what that means, what is a glycolipid? A glycolipid is a conjugate where a carbohydrate is covalently linked to a lipid. So this is the basic structure of glycolipid. So what you find, that this molecule will change, that this may be a monosaccharide, oligosaccharide or a complex oligosaccharide, whereas this will be the Y represents the ceramide unit, which I have not made here, so you can refer to this molecule. So what you have to remember is this Y is always going to be the same. What is going to differ when we will talk of classification of glycolipids? This head will change. Now let us go So now we can classify glycolipids into different categories on the basis of the alcohol which is present there, then on the basis of the fatty acid which is linked to the amino group resulting in the amide bond, and also on the number and types of carbohydrate moieties and the derivatives which are linked to an alcohol. So if we talk of alcohol, till now I have compared glycosphingolipid, that is with a broad classification with a phospholipid. But when we are talking of glycolipids, they themselves can be divided into two categories depending upon the backbone. Like, what is the backbone made up of? If the backbone is made up of sphingosine, then they are known as glycosphingolipids, and these are predominant in animals and humans. And if the backbone is made up of alcohol, which is glycerol, then these glycolipids are known as glycoglycerolipids, and they are found more frequently in plants. So when we say, in a glycolipid, when a monosaccharide or an oligosaccharide is connected to a sphingolipid, you have to remember it is called as glycosphingolipid. So when we look into the basic structure of glycosphingolipid, what you find that this ceramide bone is going to be there, and now this H is going to be replaced by a monosaccharide, a disaccharide or a trisaccharide, and it may also be a derived oligosaccharide, which will give us different categories of glycosphingolipids. But here we are talking of broad classification on the basis of the backbone of glycolipids. So in case the molecule is sphingosine, that is, you have this sphingosine, it should immediately come to your mind that it is an example of a sphingolipid. And when a monosaccharide or an oligosaccharide is connected to a glycerol that possesses fatty acids, then what we get here is we get glycerolipids. So what you find that in glycoglycerolipids, what is the backbone here? It is glycerol. Whereas in glycosphingolipids, the backbone was sphingosine. That is the difference between these two classifications, these two set of glycoglycerolipids. And then what you have to remember, that these two groups will of course be attached to fatty acid, that is, you'll get an acyl bond here, you'll get an acyl bond here. And now this OH can be attached, that is, we'll get a glycosidic bond here between a monosaccharide or a oligosaccharide. And then what we'll get is a glycoglycerolipid. So the broad classification, when we remember, what you have to remember is that for a sphingoglycolipid, you are going to make the structure of sphingosine, that is ceramide, and replace the H with a carbohydrate, the H of OH on the first carbon of ceramide. Whereas when we talk of glycoglycerolipid, then you have to remember that two of the hydroxyl groups will form a acyl linkage, and third OH will end up making a bond with a carbohydrate moiety. Now let us see how can we categorize sphingoglycolipids. So you have to remember, in sphingoglycolipids, the backbone is going to be sphingosine, and now on the type of carbohydrates which are present there, they can be cerebrosides, they can be gangliosides, or they can be lactosyl ceramides, which are also known as globosides. Whereas glyceroglycolipids has a glycerol backbone, and here we can have two categories, that is galactolipids, there the moiety will be galactose, and then you can have sulfolipids. So now let us discuss this sphingoglycolipids in detail. And now, as I've said, that we can specify glycolipids on the basis of the alcohol present in them. So the first thing which should come to your mind when we are talking of sphingoglycolipids is that the backbone is going to be sphingosine. So now where will we get? When now the second category is we'll get cerebrocytes, we'll get globocytes, we'll get gangliosides. Now globocytes, gangliosides, cerebrocytes will depend on the number and type of carbohydrate moieties and the derivatives linked to a sphingosine alcohol backbone. That is, in other words, the which groups which are attached to the first OH of the ceramide will give us different categories. So now if you look into the general structure, first of all you have to remember that when we are talking of sphingolipids, they are non-glyceride lipids. That is, glycerol is not going to be present there. So what is going to be present there? We will have a basic unit of ceramide. Please remember, inverted L, you will always have this basic unit. Now this fatty acid may also vary, and we will be discussing classification on the basis of these fatty acids also. But these are going to be long chain fatty acids. They can be 20, 24 and so on. And then where are we going to get the difference? Difference is going to be here. That is when I'm talking of cerebrocytes, globocytes or gangliosides, this H, this R group will be different here. So depending upon this R1, which I said we'll refer to this H as an R1 group, we will get different categories of glycolipids. So what we find here, this is what is the basic unit of a ceramide. That is still here. Here we have a ceramide, and I've shown you a very simple structure of a cerebrocyte. Right. So what we find that this is the sphingosine unit. Till here we have here the OH of the sphingosine or the ceramide is entering into glycosidic bond with the OH on the first carbon of the sugar unit. And what do we get? We get here is a glycosidic bond. So this is the simplest structure of a glycolipid. So this structure here, please remember, is going to be the same in all glycolipids. What is going to change? The change will come here. That is, it can be a monosaccharide, it can be a di, tri, tetrasaccharide, that is oligosaccharide, or it can also be a complex oligosaccharide. So let us see what happens next if we change these groups. So as I've already said that all glycosphingolipids have a ceramide linked with one or more sugar residues or their derivatives. So this is again what I've shown here, because I want this to be very clear that you have to keep this unit as the basic unit, right? This unit here is going to be your basic unit. This we are not going to change. This does not take part. This is not biologically active, so this does not take part in bond formation. This ends up in amide linkage. So this structure till here is going to be the basic structure. And please remember, this is the hydrophobic part of the molecule. This is the polar part and the hydrophilic part. And here now we have a sugar head. This sugar head is going to be polar, and what we'll get here is a beta linkage here. I'm sure you'll remember the alpha and the beta linkages, which I have discussed in my previous lectures on carbohydrates. So the number and type of carbohydrate moieties which will be attached to ceramide will determine the glycosphingolipid. So here I've taken an example. You have this is the simplest example of ceramide. In this ceramide, this X is going to be H. Now we can change this X. We can have different groups here. If we have, on the basis of the groups which we have there in ceramide, these can be classified into three, the glycosphingolipids: the cerebrocytes, the gangliosides, and the globocytes. So when I am talking of cerebrocytes, please remember in cerebrocyte we will just have one monosaccharide unit. It could be glucose, it could be fructose, it could be anything here. It could be galactose also, right? Now you know that glucose and galactose are epimers. Or we could have 2 or more units here. So if we have just one monosaccharide here, we call them as cerebrosides. And if we have di, tri or more, we call it as globosides. So what is the difference between a globoside and cerebroside? In cerebroside, there is going to be just one monosaccharide unit, whereas in globosides, these units can be more. They can be 2 or 3 or more. Whereas when we talk of gangliosides, then what we find that this is a complex oligosaccharide, that we will have simple sugars in that, and then we will have some heteromolecules also. For example, we can have sialic acid in it. So depending on the units of sialic acid, we will further classify gangliosides. But as of now, what you have to remember is that if we have one monosaccharide, only one sugar molecule is present, they are cerebrosides. If 2 or more sugar molecules are present, that is oligosaccharides are present, then it is globosides. And if we have a complex oligosaccharide and we have one of the members there is sialic acid, then they are going to be gangliosides. Now I want to share a fact with you, an interesting fact, that Thudikam in 1884 isolated a class of lipids from the human brain which had nitrogen and sugars, but it had no phosphorus. He named them as cerebrosides. That is, Thudikam in 1884 isolated glycolipids from cerebrosides, from the brain, and he named them as cerebrosides because they were present in the brain. And he published his work in his legendary 1884 publication, a treatise on the chemical constitution of the brain. And now we know, after his work, and a lot of work was carried out, and now we know that cerebrosides are the simplest glycosphingolipids with a single sugar linked to a ceramide. And since a single sugar is linked to a ceramide, we do not have any acidic group in it, we do not have any basic groups in it, so we say cerebrosides are neutral glycolipids. And how are cerebrosides formed? They are formed by joining of a monosaccharide connected directly to the OH at the C1 of the ceramide moiety by a glycosidic linkage. So here what you can see on your screens now is the structure. This is the structure of the ceramide molecule. We've replaced the H of a ceramide with a glucose. Now see, you already know that when an anomeric OH of the monosaccharide makes a bond with an alcohol, it results in a glycosidic linkage. And what we are finding here is we are getting a glycosidic linkage here. So this you know is the anomeric carbon, and I'm sure you remember that anomeric carbon is the carbon which is, when you are looking into the structure of the monosaccharide, which is attached to 2 oxygens. Please see, it is only this carbon which is attached to these 2 oxygens here. And then you have to remember that anomeric carbon is formed as a result of cyclization between the carbonyl group, that is, if I am taking of glucose, that is CHO on the first carbon and OH on the 5th carbon, resulting in the 6 carbon molecules which are known as anomers, that is alpha glucose and beta glucose. So, but this all I have discussed in my previous lectures, to which you can refer. But this is the glycosidic linkage here. So, glycosidic linkage is formed as a result of bond formation between the OH on the anomeric carbon and the OH of the ceramide bond, resulting in the glycosidic bond here. So, this is the basic structure of the cerebroside. So depending on what is the monosaccharide moiety present here, these cerebrosides can be classified. So now this sugar unit can either be glucose or it can be galactose. And you know that glucose and galactose are C6 sugars which are epimers, that is, which differ in configuration at one of the carbon atoms. Now, when we talk on the basis of the sugar molecule which is present in the cerebroside, if glucose is present, then that molecule is known as glucocerebroside, and if galactose is present, it is known as galactocerebroside. Glucocerebrosides are also known as glucosylceramide. Why is it known as glucosylceramide? Because we consider that to be a derivative of ceramide, that is, in which we replace the H group of ceramide with the glucose molecule. And these glucosylceramides are constituents has glucose as a constituent. As you can see here now on your screens again, that this is the basic structure is ceramide, and we've replaced the H group with the glucose molecule here. Now, where is this glucocerebroside found? It is found in non-neural tissues. It is present in liver, spleen, lungs and retina. It is a major component of skin lipids, and it plays a role in enabling water permeability barrier function of the skin. That is, it will not allow excess sweating. Now, the next class of cerebrosides is galactocerebrosides. Galactocerebrosides are also called as galactosylceramide. This is a cerebroside in which the carbohydrate constituent is galactose. So, as you can see on your screens now, here we have the ceramide unit, and in ceramide unit we have replaced, here you are getting is a glycosidic bond here, and it is known as galactocerebroside, or it is also known as galactosylceramide. Now, this galactosylceramide is present in brain and nerve myelin sheath, and it is the principal component of the brain tissue. It insulates the axons of the nerves there. So now when we talk of these cerebrosides, so the basic difference that you're going to find in these cerebrosides is that the basic molecule may be glucose. If it is glucose, then it is glucocerebroside, and if it is galactose, then it is galactosylcerebroside. And glucocerebrosides are found in non-neural cells like in spleen and liver, whereas galactocerebrosides are found in brain, in the sheaths, in the axons, and so on. So this is the basic difference. Now we also know that now the basic difference that we are going to do between these cerebrosides themselves is on the basis of the fatty acids which are present in them. And we will find out in our next lecture that there are different fatty acids, and all of them have 24 carbon atoms in them. That now the structure difference will lie in the fatty acid chain, and the galactose molecule will be present in all cerebrosides, and on the basis of the difference in the fatty acids which are attached to the amino group, that is, which are involved in amide bond, we will be able to get I will specify here 4 different categories of cerebrosides. So for this lecture, again, I had referred the books by Bruce Paula, McMurray Joe, Finar, Morrison and Boyd, and Solomon. In my next lecture, we shall be discussing the classification of cerebrosides on the basis of, that is rather when I talk of cerebrosides, galactocerebrosides, on the basis of the structure of the acid bond, acid groups, that is acyl groups, which are involved in amide linkage with the amino group, that is the structure will be different. Then we will go on to discuss acidic cerebrosides. Thank you.